Right, today I'm doing a short audio recording about viruses. Viruses are able to infect many things, from plants to plants, animals to all sorts. Um, Viruses infect plants such as tobacco plants, and this is a virus like tobacco Moscow virus (TMV), which ruins tobacco plants. Infect humans in many areas such as liver, the glands, the eyes, skins. Um, Most common ones are rashes and on the skin, such as pox and warts. Also, there's eyes, such as the conjunctivitis, and then there's keratase. Um, and there's liver, such as hepatitis, A, B, C, basically from A to E. Gastrointestinal, such as the rotavirus, and they can cause disease such as cardiac, with precardiac myocarditis, and cancer by injecting oncogenes into cells. You can get glandular fever, which is quite bad. Also, is mumps. Also, ones that attack the central nervous system, such as meningitis. Also, respiratory, such as flu, influenza, just general coughs and colds, common cold, and pneumonia. Um, the pathogenesis of viruses, they normally are like clinical, subclinical, and parent, and persistent infections are usually chronic, where a virus is detected, and they have latent, where it's ocular or cryptic, so it's hidden or obscured because there's not enough, and there's only a little bit of the genome which is replicated. There's inapparent or subclinical effect. There's known signs of presence, but it's still there, such as hepatitis C. And then the immune responses to these viruses, you can get IgG, which is late, recent, or past infections, and IgM, which is recent infection. You can tell if this is a new virus which has infected someone or an old virus. The transition can be direct through aerosol or hand-mouth, animal to animal, which could be humans accidentally, which happened with the food-and-mouth outbreak. This could be like a bite, also rabies. I mean, UK has very high quarantine, so it has managed to stop rabies, but rabies is not quite common, but occurs across across Europe, because it's one land mass, so it crosses quite easily. Whilst Britain has managed to quarantine bits going over. Also, you can get an arthropods vector, which can be in insects such as mosquitoes, leading to malaria. The classification of viruses can be family, which is family virus, and then you have subfamilies such as pox, herpes, parvo, or paravoxy. I can't say that word, sorry. Subfamilies. Also, the classification goes into whether it's enveloped or not, non-enveloped, which involves whether the genome is encased in an envelope or not. Also, classification can go into the genome, which I just mentioned slightly, with organisation and replication, and you can get types which are single-stranded DNA, RNA, double-stranded DNA, RNA. Then you can also get plus or minus stranded nucleic acid, so plus produces its own mRNA, whereas minus requires the host to produce its own mRNA through ribosomes. It has antigenic properties that can result in an immune response. The morphology / structure of viruses, you get caspid, which contains the viral nucleic acids made up of caspomers. Yes, phospholipid layer, which is the envelope which I mentioned. Also, if it has no envelope, it's also known as a naked virus. The shape is helical, or polyhedral, which can be an isohedral, which is a ball, and that's made up of 532 such domestic caposmears, which basically involves five I think it's hexagons, then which is attached to two, and that makes up a ball shape, and you get a complex shape such as smallpox, and that leads to cocky clusters and T4 bacteriophages. Culturing of the viruses can be in living cells, and you isolate viruses from host detection, such as particle antibodies. You can be fertilised in chicken eggs, which is common in laboratories, and you can have culturing in tissue culture, which can tell if it destroys local areas, specific area of a virus. Also, you can use antibiotics to stop contamination of other viruses, such as CPE, cytopathic effects. Also, you can get enzyme-linked immunosorbent assay, which checks samples for antigens. I mean, really, the definition of viruses, it's acellular, without a cellular structure. It obligates intracellular pathogens, so nearly all of the viruses must replicate using a host, such as bacteria, plant, or human. There's no independent metabolism in viruses. Also, DNA or RNA, but it can be either DNA or RNA. It can't be both. Also, they're small infectious agents. They do form viroids, which has no envelope, no cuspid, and they exist inside the cell, and they're single circular RNA, and they produce satellites which are small, between 500 and 200 nucleotides in length, and they require a helper virus, and these are most common in plants, and they lack genes for replication. And that's all.